Well, I love geeking out about everything gut related, and today I'm talking about the role of the gut microbiome in brain health. So let's get started because there's a lot to cover, and this really is just scratching the surface. So the learning objectives for today are to review the gut-brain axis fundamentals, to gain insight into how substances produced by gut microbes affect the brain, for better or worse, identify specific microbes on the GI map test that can influence brain health, again, positively or negatively, discover how alterations in short-chain fatty acids and bile acids can impact brain health, and how the stool omics add-on test can help identify these imbalances. And finally, we'll have a Q&A at the end. So what is the gut-brain axis? The gut-brain axis is a complex bidirectional communication network connecting the gut and the brain. Now, due to this bidirectional nature, the brain can influence gut function, and the gut can also influence brain function. In other words, the brain talks to the gut, and the gut talks back. Because signals pass both ways between the gut and brain, the health or disease in one can affect the other. In fact, the gut is often called the second brain because it houses a complex network of neurons called the enteric nervous system. Now, the enteric nervous system, which lines the GI tract, contains over 500 million neurons. That's actually more neurons than the spinal cord or peripheral nervous system. And it communicates using a variety of neurotransmitters and signaling molecules. The gut and brain are in constant communication, and the enteric nervous system plays a role in this communication. The gut can communicate with the brain through multiple pathways. For example, the vagus nerve directly transmits sensory information from the gut to the brain, while the bloodstream carries signaling molecules and metabolites produced in the gut to the brain. So the vagus nerve is really heavily involved in gut-brain communication. Now, the vagus nerve is one of the 12 cranial nerves. It originates from the medulla of the brainstem and travels down through the body, branching out along the way. It's composed of 80% afferent neurons, which carry information from the gut to the brain, and 20% efferent neurons, which carry information from the brain to the gut. This simply means that the vagus nerve primarily carries information from the gut to the brain. Simply put, the gut talks to the brain more than the brain talks to the gut. The gut microbiota, the organisms living in the gut, are a pivotal component of the gut-brain axis. That's because gut microbes produce or stimulate the production of many substances that convey messages between the gut and brain. Alterations in the composition of the gut microbiota, known as gut dysbiosis, have been associated with gut-brain axis dysfunction and even neurodegeneration. Because the state of the gut microbiota significantly impacts the brain, if these microbes become imbalanced, the brain can suffer. Here's a great review article from 2023 that states, intestinal dysbiosis leads to a change in the bidirectional relationship between the central nervous system and gut microbiota, which is linked to the pathogenesis of neurodevelopmental and neurological disorders. Increasing preclinical and clinical studies/evidence indicate that gut microbes are a potential susceptibility factor for the progression of neurological disorders, including Alzheimer's disease, Parkinson's disease, multiple sclerosis, and autism spectrum disorder. So brain-related symptoms that may occur as the result of gut dysbiosis include anxiety, depression, ADHD, migraine, mood and behavioral issues such as irritability or aggression, cognitive issues like brain fog or memory problems, and food cravings. Neurological disorders associated with gut dysbiosis include Alzheimer's, Parkinson's, MS, and autism. So let's talk about some of the ways that the gut microbiome affects the brain. Substances produced by gut microbes directly or produced by the body in response to gut microbes influence the brain, for better or worse. In this webinar, we're going to focus on microbial metabolites, that's substances produced by gut microbiota. that can exert beneficial or detrimental effects on the brain. In studies, some of the microbial metabolites that have been found to impact the brain are neurotransmitters and hormones, short-chain fatty acids, bile acid, lipopolysaccharide, better known as LPS, hydrogen sulfide, histamine, acetaldehyde, which is produced by candida, ammonia, D-lactic acid, and P-creosol. And that's just some of the major ones. There are more. Although we don't have time to cover all of these today, I will be discussing a handful of them in greater detail. Some of the key microbes that produce these metabolites and the metabolites themselves can be directly measured on stool tests, such as the GI map test plus stool omics add-on panel, which is pretty new from Diagnostic Solutions Laboratory. This would include microbes that produce neurotransmitters and hormones, short-chain fatty acids, secondary bile acids, LPS, histamine, hydrogen sulfide, and acetaldehyde, as well as the microbial metabolites, short-chain fatty acids, and bile acids. The levels of other metabolites that are not directly measured can be estimated based on the amounts of gut microbes detected on the test that are capable of producing these metabolites. This would include LPS, histamine, hydrogen sulfide, acetaldehyde, and others. So let's start to dig in a little bit deeper and talk about some of the main microbial metabolites that can impact the brain, beginning with neurotransmitters and hormones. A number of gut microbes can either produce neurotransmitters directly, or they stimulate intestinal cells to produce neurotransmitters. This includes serotonin, dopamine, norepinephrine, and GABA. Altered levels of these neurotransmitters have been linked to a variety of neurological and mental health conditions, since they play crucial roles in regulating mood, motivation, attention, and other brain functions. In fact, over 90% of the body's serotonin is produced in the gut. The majority is produced by the enteromassin cells in the gut lining. However, some species of bacteria can directly synthesize serotonin. And more than 50% of dopamine in the human body is synthesized in the gut. Although gut-derived versions of these neurotransmitters do not cross the blood-brain barrier, they can still influence the brain. Gut microbiota can stimulate the vagus nerve, and the stimulation can alter the levels of neurotransmitters in the brain. So, for example, while gut-produced serotonin doesn't directly enter the brain, it activates the vagus nerve, transmitting signals to the brainstem, which then integrates these serotonergic signals and projects them to higher brain regions where they do influence mood, cognition, and stress responses. Gut-produced serotonin also appears to alter the expression of serotonin receptors in the brain and affect blood-brain barrier permeability. Furthermore, there are other ways that the gut can impact brain levels of serotonin. The gut affects the availability of tryptophan, a serotonin precursor, which does cross the blood-brain barrier to be used by the brain cells to produce serotonin. Tryptophan is an amino acid that must be obtained through the diet because the human body can't synthesize it. For the body to effectively use tryptophan, it must be properly digested and absorbed. As a result, good protein digestion and small intestinal absorption are required to ensure that tryptophan is available for the brain. And gut inflammation resulting from dysbiosis can cause increased activity of the IDO enzyme, which diverts tryptophan away from the serotonin pathway and towards the kynurenine pathway. This leads to the production of kynurenic acid, which can be further metabolized into quinolinic acid, which is a neurotoxic compound that can contribute to neuroinflammation and neurodegeneration. Elevated quinolinic acid has been associated with mood disorders, Alzheimer's, and Parkinson's. Now, when it comes to hormones, the hormones GLP-1 and PYY are produced by L cells in the gut and released in response to microbial metabolites, such as short-chain fatty acids and secondary bile acids. These peptides affect mood, memory, and learning, GLP-1 has been shown to enhance hippocampal synaptic plasticity, improve learning, memory, and motor function, as well as mitigate glial cell activation, suggesting potential neuroprotective effects in neurodegenerative disorders, such as Alzheimer's and Parkinson's. These gut-produced peptides can stimulate the vagus nerve and cross the blood-brain barrier to directly impact the brain. Here's what to look for on functional lab tests. On the GI map test, look for low commensal/keystone bacteria. Some of these bacteria produce neurotransmitters, while others stimulate the intestinal cells to make neurotransmitters. On the organic acids profile, look for low or high serotonin metabolites, that is 5-HIAA, low or high dopamine metabolites, that'd be the marker HVA, low or high norepinephrine-epinephrine metabolites, that's VMA, and high quinolinic acid. Next, we have short-chain fatty acids. Short-chain fatty acids are produced by bacteria in the colon when they ferment dietary fibers and resistant starch. Humans lack the enzymes to degrade the bulk of dietary fibers, so these non-digestible carbohydrates pass through the upper GI tract unaffected and are fermented in the large intestine by colonic microbiota. Major factors that can impact short-chain fatty acid production include dietary fiber intake, gut inflammation, and colonic transit time. The amount and type of fiber consumed has dramatic effects on the composition of the gut microbiota and consequently on the type and amount of short-chain fatty acids produced. A diet low in fermentable fibers will cause reduced levels of short-chain fatty acids. Inflammation can inhibit the growth of many key short-chain fatty acid-producing species. Rapid transit time, or you might see diarrhea, can reduce the time available for fermentation, limiting short-chain fatty acid production. The three major short-chain fatty acids made by the gut microbiota are butyrates, acetate, and propion, which account for approximately 90 to 95% of those produced in the gut. Acetate accounts for around 50 to 60% of the total short-chain fatty acids, followed by propionate at 20% and butyrate at 20%. Other short-chain fatty acids like valerate are present in smaller quantities. Now, following their production, short-chain fatty acids are absorbed by colonocytes, the colon cells, which largely depend on butyrate for their energy supply. The remaining 5% are excreted in the stool, which we can measure. Those not consumed by the colonocytes are transported from the colon into the bloodstream and are taken up by organs where they act as substrates or signaling molecules. Although only a minor fraction of colon-derived short-chain fatty acids reach the bloodstream, these short-chain fatty acids have a significant impact on various organs and systems, including the brain and central nervous system. Short-chain fatty acids can influence brain function directly or indirectly. Because they are able to cross the blood-brain barrier, they are able to have a direct effect on the brain. But they also stimulate the production of other substances like GLP-1 and PYY that affect the brain. Short-chain fatty acids have a wide range of effects on the brain. They stimulate the vagus nerve, which can influence brain function and inflammation. They improve the integrity of the blood-brain barrier by increasing the expression of tight junction proteins that seal the gaps between cells. Short-chain fatty acids decrease permeability of the blood-brain barrier to prevent harmful substances from entering the brain. Disruption of blood-brain barrier integrity is implicated in the pathology and progression of neurodegenerative disorders. Short-chain fatty acids also promote neurogeneration. They can cross the blood-brain barrier and promote neurogenesis, which is the process of generating new neurons, which is important for brain health and plasticity. They decrease neuroinflammation through a number of mechanisms, including shifting microglia towards a more anti-inflammatory state, reducing the production of pro-inflammatory cytokines, and increasing the levels of brain-derived neurotrophic factor. Now, speaking of brain-derived neurotrophic factor, or BDNF for short-chain fatty acids, increase BDNF, which is a key protein involved in learning and memory that has also been shown to decrease neuroinflammation. Some clinicians have actually referred to BDNF as miracle grow for the brain. Low BDNF levels have been linked to mood disorders and cognitive decline. Short-chain fatty acids also activate AMPK. This is an enzyme involved in energy metabolism and cellular metabolism. In the brain, AMPK is involved in synaptic transmission and plasticity, playing a role in memory and learning. It can also have protective effects against neuronal dysfunction and neuronal death, particularly in situations like ischemia and neurodegenerative diseases. AMPK dysregulation has been reported in neurodegenerative diseases such as ALS, Alzheimer's, and Parkinson's. Another thing that short-chain fatty acids do is they regulate the normal development and function of microglia. Microglia, which are the immune cells of the central nervous system, can be broadly classified into M1 and M2 phenotypes based on their function and morphology. M1 microglia are pro-inflammatory, contributing to neurotoxicity, while M2 microglia are anti-inflammatory. M1 microglia are implicated in the pathogenesis of neurodegenerative diseases, like Alzheimer's and Parkinson's. Microglia can transition between M1 and M2 phenotype based on environmental cues and stimuli. Short-chain fatty acids have been shown to promote M2 polarization and inhibit M1 activation in microglia. As I mentioned already, short-chain fatty acids promote the secretion of GLP-1 and PYY, peptides that affect mood, memory, and learning. Short-chain fatty acids promote the production of serotonin in the gut as well. Again, although these gut-produced serotonin does not cross the blood-brain barrier, it can impact the brain by stimulating the vagus nerve. Another thing short-chain fatty acids do is they inhibit histone deacetylases, or HDACs. HDACs are enzymes that remove acetyl groups from histone. While HDAC activity plays a crucial role in regulating gene expression and other cellular processes, increased HDAC activity can be detrimental, contributing conditions like cancer and neurodegeneration. Increased HDAC activity has been found in Parkinson's, schizophrenia, depression, and dementia. Among the short-chain fatty acids, butyrate is the most potent HDAC inhibitor. Additionally, excessive activity of HDAC enzymes can negatively impact the blood-brain barrier. Increased HDAC activity can lead to changes in tight junction proteins, increasing blood-brain barrier permeability. And HDAC enzymes can increase the activity of NF-kappa B, a transcription factor that, if excessive, can contribute to neurodegeneration by promoting inflammation, oxidative stress, and exotoxicity. NF-kappa B activation has been linked to several neurodegenerative disorders, including Alzheimer's. In Alzheimer's, NF-kappa B is thought to contribute to amyloid plaque formation, glial cell activation, and neuronal death. Finally, short-chain fatty acids activate G protein-coupled receptors, which can reduce neuroinflammation by modulating glial cell function, protect neurons from apoptosis, and maintain blood-brain barrier integrity. So here's what to look for on functional lab tests. On the GI map, again, look for low commensal keystone bacteria in general, but also for low levels of some key short-chain fatty acid-producing microbes. On this slide, I've included a list of some key acetate, propionate, and butyrate producers. Although the GI map doesn't measure some of these species individually, most are included in the two phylum markers. Acetate and propionate are mainly produced by Bacteroidetes, whereas butyrate is largely produced by Firmicutes, in particular Clostridia. On the report, the three main markers that represent butyrate producers are Faecalibacterium prosnitz, Roseburia, and the Firmicutes phylum. Although not all species within the Firmicutes phylum produce butyrate, most butyrate producers do belong to this phylum. On the other hand, Bacteroidetes are more associated with the production of acetate and propionate. Now, some species are capable of producing more than one type of short-chain fatty acid, such as Akkermansia and many Bacteroides species, which can make both acetate and propionate. On the stool Omex add-on panel, look for low major straight-chain fatty acids or low percentage of acetate, butyrate, propionate, or valerate. One thing to note, however, is that although we're primarily concerned about low levels of short-chain fatty acids, high levels of some short-chain fatty acids can also be problematic. Some short-chain fatty acids can have both positive and negative effects on the brain, particularly propionate and valerate. Excess propionate can contribute to various neurological issues, including neuroinflammation, altered neural circuitry, reduced social interaction, anxiety-like behaviors, and abnormal motor movement. High propionate levels have been seen in patients with autism and dementia, especially Alzheimer's disease. Remember, bacteria in the Bacteroidetes phylum are the primary producers of propionate in the gut. Then excess valerate can disrupt the blood-brain barrier and trigger neuroinflammation. Increased valerate may play a role in the initiation and progression of Alzheimer's disease. Next, we'll move on to bile acids. Bile acids are synthesized and conjugated in the liver, then stored in the gallbladder and released into the duodenum when food is ingested. In the colon, they are modified by bacteria into secondary bile acids. Approximately 95% of the bile acids released into the gut are reabsorbed in the ileum, which is the last part of the small intestine, and transported back to the liver through the portal vein. However, a small amount of the bile acids in the enterohepatic circulation, up to 10%, escape to the systemic circulation, from where they can be taken up into the brain by passive diffusion or by active transport through the blood-brain barrier. Although bile acids may be synthesized locally in the brain, the majority of brain bile acids are taken up from the systemic circulation. While the primary bile acids, cholic acid, which we often abbreviate as CA, and chenodeoxycholic acid, which is CDCA, can be synthesized in the brain, the bile acid composition in the brain is highly complex, consisting of both primary and secondary, unconjugated and conjugated bile acids. The most abundant bile acids in the brain are CDCA, DCA, and CA. Bile acids activate receptors in the brain, such as FXR and TGR5. While bile acid activation of FXR is important to maintain normal cognitive function and motor coordination, activation of TGR5 induces key anti-inflammatory pathways. Like with short-chain fatty acids, it is important to have a proper balance of the different types of bile acids. Maintaining a balanced composition of bile acids is crucial, as certain types at elevated levels can negatively impact brain function. Altered bile acid profiles have been observed in neurological disorders. While some bile acids have more neuroprotective effects, others are more cytotoxic. UDCA and PDCA tend to improve brain-related conditions, so low levels of those may be problematic, while BCA and LCA may worsen brain-related conditions if present in excessive amounts. The cytotoxicity of bile acids is generally correlated with their hydrophobicity. More hydrophobic bile acids tend to be more cytotoxic, while less hydrophobic bile acids are less toxic. LCA has the highest hydrophobicity, followed by DCA, then CDCA, then CA, then UDCA. The hydrophilic bile acid, UDCA, and its taurine conjugated derivative, TUT, have low toxicity and protective properties. When present at high levels, DCA and LCA, as well as GDCA and TDCA, which are conjugated forms of DCA, can be neurotoxic and have been implicated in Alzheimer's, Parkinson's, and MS. DCA can induce mitochondrial dysfunction and oxidative stress in neurons, increase blood-brain barrier permeability, and activate neuroinflammatory pathways like toll-like receptor 4 and NF-kappa B. In Alzheimer's disease, researchers have observed increased levels of the secondary bile acid DCA and decreased levels of the primary bile acid CA. In fact, in one study, an increased ratio of DCA to CA was strongly associated with cognitive decline. Now, LCA can activate microglia, leading to neuroinflammation, disrupt calcium homeostasis, endoplasmic reticulum stress, and damage mitochondrial cell membrane. GDCA and TDCA are less toxic than DCA, but still associated with cognitive decline. They can cross the blood-brain barrier and trigger low-grade inflammation. So the synthesis, absorption, and secretion of bile acids is tightly regulated to ensure a constant bile acid pool and to prevent cellular toxicity. Bile acid pool size and composition may be altered by diet, dysbiosis, or liver-related disease. Dysregulation of bile acid synthesis or metabolism can affect brain function. Bile acids affect the brain in numerous ways. Like short-chain fatty acids, they can impact the brain through both direct and indirect mechanisms. They stimulate the vagus nerve to send signals to the brain that influence brain function. They activate receptors in the brain. By binding to receptors in the brain, bile acids made in the gut that cross the blood-brain barrier, as well as bile acids produced in the brain itself, regulate brain activity and function. Bile acids, at least some of them, have neuroprotective effects. Ka and its conjugates TUDCA and GUTCA have demonstrated neuroprotective properties, particularly in reducing neuronal apoptosis, oxidative stress, inflammation, and endoplasmic reticulum stress. These bile acids are actually being investigated as potential therapeutic agents for a variety of neurological, neurodegenerative, and neuropsychiatric disorders, including Alzheimer's, Parkinson's, and depression. Bile acids also modulate the blood-brain barrier. Disruption of the blood-brain barrier is a hallmark feature of many neurological disorders. Certain bile acids, like UTCA, can protect the blood-brain barrier and reduce its permeability. However, hydrophobic bile acids, like DCA and CDCA, can increase blood-brain barrier permeability by disrupting tight junctions. At high concentrations, bile acids can also act as detergents to dissolve the lipid layer of the blood-brain barrier. So normal concentrations of bile acids can decrease blood-brain barrier permeability, but elevated amounts of bile acids, DCA and CDCA in particular, can increase blood-brain barrier permeability, triggering neuroinflammation and neuronal dysfunction. Now, bile acids also promote the secretion of GLP-1 and PYY from L cells in the gut, which affect mood, memory, and learning. These gut-produced peptides may reach the brain via the systemic circulation or through the vagus nerve. Bile acids promote the production of serotonin as mentioned earlier, although gut-produced serotonin does not cross the blood-brain barrier, it can impact the brain by stimulating the vagus nerve. Bile acids also influence the production of BDNF. This is a protein that's to the formation of memory and cognitive function. Low BDNF levels have been linked to mood disorders and cognitive decline. TUDCA has been shown to increase BDNF levels, whereas LCA, DCA, and CDCA appear to downregulate BDNF expression. They exhibit both antibacterial and antifungal properties, which means they can prevent certain microbes from overgrowing in the gut. Because bile acids have both direct and indirect antimicrobial effects on gut microbes, having low levels of bile acids in the intestinal tract can lead to bacterial and yeast overgrowth. The overgrowth of these microbes can then have negative effects on the brain. For example, an overgrowth of gram-negative bacteria can cause high levels of LPS, which is lipopolysaccharide. And LPS produced in the gut can cross the blood-brain barrier and cause neuroinflammation. And an overgrowth of candida can lead to high levels of acetaldehyde, which is neurotoxic. Furthermore, candida can also react with dopamine to form sulfonol, a neurotoxic compound which has been implicated in the pathogenesis of Parkinson's. Finally, bile acids affect the absorption of fat-soluble vitamins. In the gut, bile acids act as emulsifiers of dietary fat, breaking down large fat globules into smaller droplets, which makes them more accessible to digestive enzymes. Without bile acids, fat-soluble vitamins cannot be properly absorbed, leading to deficiencies. Because fat-soluble vitamins A, D, E, and K are vital for brain health, deficiencies in these vitamins can have negative consequences for the brain, potentially impacting cognitive function and increasing the risk of neurological disorder. Here's what to look for in functional lab tests. On the GI map, look for low levels of bacteria that help convert primary bile acids to secondary bile acids. The conversion of primary bile acids to secondary bile acids in the colon involves a two-step process. First, deconjugation by bile salt hydrolase, which is BSH, and second, 7-alpha dehydroxylation. Having a lack of the bacterial species that express BSH, which removes the glycine or taurine conjugation from primary bile acids, or 7-alpha dehydroxylase, which allows bacteria to convert primary bile acids to secondary bile acids, can lead to lower levels of some of the most brain-protective secondary bile acids, like UDCA and TUDCA, and lead to higher levels of primary bile acids, like CDCA, which can be harmful to the brain at excessive levels. Also look for dysbiosis on the GI map report. Having an imbalance of bacteria can alter the composition of the bile acid pool, resulting in higher levels of more harmful secondary bile acids, like LCA and DCA, and lower levels of more neuroprotective secondary bile acids. That is, according to researchers, the reported increases in the levels of the secondary bile acids within the brain during neurodegeneration most likely are a result of an altered gut microbiome producing higher levels of these bile acids, which subsequently enter the brain. On the stool Omex add-on panel, look for low or high total bile acids. Look for poor conversion of bile acids, which you'll see as a high primary bile acids percentage and low secondary bile acids percentage. And look for abnormal bile acid percentages of the individual primaries and secondaries. Next, let's talk about lipopolysaccharide, or LPS. Lipopolysaccharide, also known as endotoxin, is a major component of the outer membrane of gram-negative bacteria. Gram-negative bacteria, and thus also LPS, are naturally present in the gut. LPS is released from these bacteria both during normal metabolic processes, like cell division and shedding of outer membrane vesicles, and when they die or are killed. The effects of LPS on the brain are highly dose-dependent. Low levels may be beneficial, as LPS plays a role in maintaining immune balance and gut homeostasis, while high amounts can be harmful and cause inflammation. When gram-negative bacteria overgrow in the gut, excessive amounts of LPS are released. LPS interacts with toll-like receptors, primarily toll-like receptor 4, to trigger pro-inflammatory reactions. High levels of LPS can cause systemic inflammation and neuroinflammation. In fact, LPS administration is frequently used to study neuroinflammation-associated diseases in experimental models. Like short-chain fatty acids and bile acids, LPS can impact the brain through both direct and indirect mechanisms. LPS can send signals to the brain through the vagus nerve, as well as enter the bloodstream and cross the blood-brain barrier to bind to receptors on brain cells. High amounts of LPS can be harmful to the brain because it stimulates the vagus nerve, which can lead to neuroinflammation. It causes leaky gut because it can stimulate the release of zonulin in the gut. When zonulin levels are high, it causes tight junctions to loosen, leading to increased intestinal permeability. This allows the LPS and other potentially harmful substances to enter the bloodstream. It causes leaky brain. So not only leaky gut, but leaky brain as well. LPS can disrupt the tight junctions in brain endothelial cells, leading to increased blood-brain barrier permeability. Blood-brain barrier disruption has been associated with a wide range of brain disorders and conditions, including Alzheimer's, Parkinson's, and MS. It induces neuroinflammation. LPS binds to toll-like receptor 4, which leads to the production of pro-inflammatory cytokines. It reduces BDNF. LPS-induced inflammation can decrease BDNF. Some common symptoms associated with low BDNF include depression, anxiety, difficulty learning and remembering, and problems with attention and focus. Decreased BDNF has been implicated in neurodegenerative diseases like Alzheimer's and Parkinson's. It activates microglia, astrocytes, and dendritic cells. LPS-induced inflammation causes inappropriate action of these cells. When stimulated by high amounts of LPS, microglia change into an inflammatory phenotype that promotes expression of pro-inflammatory mediators. It stimulates apoptosis of neuronal cells. LPS-related toll-like receptor 4 activation causes extensive neuronal cell death. LPS contributes to the formation of amyloid plaque, a hallmark of Alzheimer's disease. LPS can enhance the production of amyloid beta peptides, which are the primary components of amyloid plaque, as well as impair the clearance of these peptides from the brain, leading to their accumulation and aggregation into plaque. Finally, LPS damages myelin, the insulating sheath around nerve fibers. Myelin damage has been implicated in both Alzheimer's disease and multiple sclerosis. On the GI map, look for high levels of LPS-producing bacteria. This includes Escherichia, Enterobacter, Morganella, Pseudomonas, Citrobacter, Klebsiella, and Proteus. Finally, we have hydrogen sulfide. A number of gut microbes are able to produce hydrogen sulfide gas, and this gut-produced hydrogen sulfide can enter the bloodstream and cross the blood-brain barrier to influence brain function. Some gut bacteria break down cysteine, a sulfur-containing amino acid, to produce hydrogen sulfide as a byproduct. Others produce hydrogen sulfide when they reduce sulfate. Hydrogen sulfide can have both protective and harmful effects on the brain, depending on its concentration. In other words, too little or too much hydrogen sulfide is detrimental to brain health, making hydrogen sulfide a double-edged sword. Hydrogen sulfide has beneficial effects at physiological concentrations, but harmful effects at excessive concentration. Altered hydrogen sulfide levels have been associated with memory problems, difficulty concentrating, brain fog, headache, anxiety, depression, and neurodegenerative disorders. When hydrogen sulfide is present in the brain at normal concentrations, it acts as an anti-inflammatory, antioxidant, and neuroprotective agent, protecting neurons and synapses from abnormal pathology. Hydrogen sulfide actually appears to function as a significant cellular antioxidant. It also upregulates glutathione synthesis. Hydrogen sulfide enhances the activity of NMDA receptors, to induce long-term potentiation in the hippocampus, which plays a role in memory formation. Hydrogen sulfide also plays a role in regulating cerebral blood flow by causing vasodilation in cerebral blood vessels. This vasodilation increases blood flow to the brain, providing increased delivery of oxygen and nutrients needed for neuronal function. Hydrogen sulfide helps maintain the integrity of both the intestinal barrier and the blood-brain barrier. Unfortunately, elevated levels of microbiota-derived hydrogen sulfide can compromise gut barrier integrity, leading to a leaky gut and the release of pro-inflammatory substances, such as LPS, into systemic circulation. One of the ways that excessive hydrogen sulfide is by interfering with the colon's ability to use butyrate, a short-chain fatty acid. And because high levels of hydrogen sulfide also disrupt the blood-brain barrier, it can then allow those substances to enter the brain. The breach in the blood-brain barrier is a critical factor in neurodegenerative diseases like Alzheimer's and Parkinson's, where neuroinflammation accelerates the degeneration of neurons. At physiological levels, hydrogen sulfide exerts anti-inflammatory effects on glial cells, limiting excessive microglia activation and promoting the resolution of inflammation. However, elevated hydrogen sulfide levels overactivate microglia and astrocytes, causing a sustained inflammatory environment in the brain, leading to progressive neuronal loss. Again, hydrogen sulfide is quite the Jekyll and Hyde. Hydrogen sulfide increases the levels of neurotransmitters such as serotonin, dopamine, epinephrine, norepinephrine, GABA, and glutamate. This means that altered hydrogen sulfide levels can lead to altered neurotransmitter levels. Normal doses of hydrogen sulfide are beneficial to mitochondria, whereas higher doses inhibit mitochondrial respiratory enzymes and increase the production of ROS, leading to oxidative stress, mitochondrial dysfunction, and neuronal injury. Altered hydrogen sulfide levels have been implicated in various neurological conditions, with both excessive and deficient hydrogen sulfide levels potentially contributing to pathogenesis. Although research has shown that the hydrogen sulfide levels are dysregulated in individuals with mood disorders and neurodegenerative disorders, there are conflicting results in studies as to whether the levels are too high or too low. This is where we have to keep in mind that both low and high levels of hydrogen sulfide negatively impact the brain. And some researchers believe that redistribution between hydrogen sulfide pools in the body may be a critical determinant in the pathological consequences. For example, both high and low levels of hydrogen sulfide have been linked to Alzheimer's disease pathology. Decreased and increased plasma hydrogen sulfide levels have been noted in Alzheimer's patients, but brain hydrogen sulfide levels are typically reduced. In one study, the plasma hydrogen sulfide level was negatively correlated with the severity of the disease. When it comes to Parkinson's, there may be low levels of hydrogen sulfide in the plasma and brain, but a significantly higher level of hydrogen sulfide producing bacteria, especially the sulfovibrio, in the gut. Again, numerous bacteria in the gut have the capacity to produce hydrogen sulfide via cysteine degradation. This includes Fusobacterium, Escherichia coli, Klebsiella, and Citrobacter. Hydrogen sulfide is also produced to a lower extent by sulfate-reducing bacteria. This would include the sulfovibrio species. On the GI map, look for low or high levels of hydrogen sulfide-producing bacteria. We would not want to see low levels, that is less than DL on the report, for any commensal hydrogen sulfide producers, like Bacteroides fragilis. We would also not want to see high levels of commensal or opportunistic hydrogen sulfide producers. So with all this information in our head, how can we help our clients with brain-related issues? First, we can utilize stool testing to identify gut microbiome imbalances. The primary test here would be the GI map test, but consider the stool omics add-on panel to test the short-chain fatty acids and bile acids. Then on the test report, we can look for altered levels of gut microbiota and microbial metabolites that have been associated with that client's brain-related symptoms or condition in the research. For example, some of the imbalances we may see in a patient suffering from major depressive disorder are a reduced level of butyrate-producing bacteria, a lower level of bifidobacterium and lactobacillus, an overgrowth of Enterobacteriaceae and or proteobacteria. These are groups of bacteria that produce LPS, an overgrowth of Klebsiella. Now, what you need to know about Klebsiella is they are super producers of histamine, as well as LPS producers. Plus, the species Klebsiella aeruginosa has been shown to degrade estradiol to cause lower estrogen levels in women with depression. An overgrowth of Morganella is also seen with depression. Besides being another super producer of histamine and an LPS producer, Morganella creates inflammatory compounds which activate immune responses that contribute to depression. Altered short-chain fatty acids have also been seen with depression. Again, particularly decreased CDCA and increased LCA. Once we know what microbes or metabolites are present at altered levels, we can create a protocol that addresses and corrects these imbalances. This may include low commensal bacteria, high LPS producing bacteria, lower high bile acids, impaired bile acid conversion, low short-chain fatty acids, or the other things we discussed during this webinar. Unfortunately, there just isn't enough time today to talk about how to specifically correct each of these issues. Now, if you'd like to learn more about the lab tests mentioned during this webinar, you can visit the Diagnostic Solutions Laboratory website to investigate gut imbalances that may be negatively affecting the brain. I highly recommend the GI map test plus stool omics add-on. The organic acids profile may also be helpful to identify issues that may be negatively affecting the brain. On the website, you can download a copy of the GI map interpretive guide to learn more about how to interpret the markers on both the GI map X report. And if you need more guidance on interpreting GI map, stool omics, or organic acid tests for your clients, give the lab a call to schedule a clinical consultation with the medical education team. And if you're interested in learning more from me, you can visit my website at pomeroyinstitute.com, where I offer one-on-one practitioner mentoring sessions on lab test interpretation, as well as comprehensive practitioner training courses. Now, in honor of this webinar, you can use the coupon code RUPA, JUN for June 25, to receive a $250 off my in-depth GI map interpretation course called GI Pathogens and Dysbiosis using the GI map in clinical practice. And this coupon code is good through July 7th.
Welcome to Rupa Health, the best place to order, manage, and track results from over 30 different lab companies in one single place for free. It's going to take you under 2 minutes to sign up, and you can order any functional medicine lab for your client in under 30 seconds. Let me show you how it's done. So here's our beautiful interface. I'm typing in the name of my client, selecting the lab that I want to order for them, and hitting send. From there, Rupa and their amazing team handle the rest. They email the client, collect payment, and even offer an interest-free 3-month payment plan. We've also built the world's largest library of information about chronic health conditions, the lab tests that can help you find the root cause, and the evidence-based interventions that you can use to help people heal from them. It's called the Rupa Health Magazine. There, we have in-depth articles about almost any health condition you can imagine. And we give you step-by-step protocols that other clinicians have used to help their clients heal and that are verified by evidence-based sources. You should totally check it out, and it'll transform your practice. And we can't wait to see you. So make sure you sign up today at rupahealth.com.